Hello everyone. In this video we are going to discuss a family of viruses called the retroviruses. Before going to do that, let's first remind ourselves what is transcription. So as you know, transcription is the process through which the information encoded in the DNA are used to form the RNA molecule. So this is a transcription process. And as you can see, the information encoded here in DNA are used to make a copy of RNA molecule. And as we said, in many cases, this is going to be followed by a translation process. Okay, so this is transcription. And if we go in the reverse direction, this way, from an RNA molecule to a DNA molecule, this is going to be called reverse transcription. Okay, so the enzyme allowing us to do this reverse transcription is found in some viruses, and an example of family of viruses that has this enzyme is the retroviruses. So they can go in this opposite direction or in this backward direction. See the name retro. So retro means something like backward. So they can move backward, moving the, I don't mean movement, but I mean they can reverse transcribe the RNA. So the information that is encoded in the RNA can be reverse transcribed, and you get the DNA code from the RNA code. So the enzyme allowing this is the reverse transcriptase enzyme. And this family of viruses are characterized by having this reverse transcriptase enzyme. Okay. One example of this family is the HIV virus. So HIV, as you know, is a human immune deficiency virus. It is the virus responsible for causing the AIDS, the acquired immune deficiency syndrome, and again, it is an RNA virus. And as we have seen in the influenza virus, it gets this membrane shown here in green from a prior host cell. You remember when the virus exits the host cell, usually it takes the cell membrane as a membrane for the virus. So it is derived from the host cell membrane, as you can see here. And again, it is an RNA virus. You have two copies of the RNA genome. So this RNA shown here in red, we have two copies of the RNA genome, and we have a group of proteins, or actually glycoproteins. That's why they are referred to as GP. GP means glycoprotein. We have many of them on the surface of the virus. The most important among them is the GP120, because this is the one that will direct the virus to the host cells, and the host cell targeted by the HIV are the ones having the CD4 receptors in our body. So this is a type of T lymphocytes that are very essential in our immune system. So unfortunately, the HIV targets this very specific type of cells, which is a key player in our immune system. So by disabling these cells, the capacity of the immune system to fight against disease will drop dramatically, and that's why usually the AIDS disease will end up in most cases by the death of the individual, because now without most of the capacity of the immune system to fight against diseases, the person can get ill and die even from normal bacterial or fungal infection that a healthy individual can very easily survive and overcome. In addition to the GP120, we have the enzymes protease and integrase and reverse transcriptase. So reverse transcriptase, as you remember, is the enzyme that will help this RNA genome to be reverse transcribed into DNA. Then the integrase will allow this genome to be integrated, the converted one, which is now a DNA, to be integrated into the host cells. And protease is the enzyme that cuts the protein into smaller fragments. So let's see how the components, these different enzymes, will work during the infection caused by the HIV. So the virus HIV will target host cells. Host cells that will be targeted are the ones with the CD4. So the CD4 is the receptor for the virus. It will bind to the GP120 on the surface of the virus. Now the virus will be internalized like that. So now the virus is internalized by endocytosis. Once inside the host cell, the virus will use the reverse transcriptase enzyme, so converting the RNA genome into a DNA genome. So now the virus has a DNA genome inside the host cell. So what will happen next? The second enzyme will come to play, which is the integrase. So this integrase will integrate this viral DNA genome within the host DNA. So the host cell chromosome is now being changed. It's now being edited by the insertion of this foreign viral DNA. Now the virus is hijacking the host cell. It is making new viral RNA molecules. These viral RNA will be translated inside the host to make viral proteins. So imagine these viral proteins, big chunk of viral proteins. Then the protease enzyme will come to cut these long proteins into smaller fragments. Then assembly will happen and the virus will leave the host cell, taking part of the cell membrane with it. And now this mature virion, or actually many virions, will come out of the cell after now that the DNA of the cell is being changed completely. And this is a complete and permanent change. So please keep in mind that this change is not transient, as we have discussed with proviruses. Do you remember when we discussed lysogenic viruses that may infect bacteria and integrate as a provirus? But in some cases, this provirus can be excised out of the chromosome. So the case with the HIV is different, because this virus that has reverse transcribed its genome into a DNA is now going to be integrated forever. It is not a transient integration. So the change of the host cell DNA is permanent, and because of that, this cell will no longer function as a immune cell. And as you can see, many virions will come out to attack even more and more host cells. And as we said, this is a major immune player in our body, and because of that, the immunity will drop dramatically, leading in most cases to death. So this, in summary, summarizes the whole process of the HIV and how the pathogenesis is caused by this virus.